we have looked at the different composition of tissues and cells in the airways, which is trachea, bronchus, bronchiole, and alveoli. So I want you to check, right? There's this common structure that we find in certain airways. So it's easy for you to remember. So you see, whenever you have goblet cell secrete mucus. So therefore, there will also be mucus gland. Please pay attention to the spelling of the word mucus gland, M-U-C-O-U-S, okay, mucus gland. And then there will also be cartilage. So these three structures will appear together in the airways, right? Goblet cell, mucus gland, as well as cartilage. Three of them will be found in the trachea and bronchus. So after that, you will no longer find goblet cell, mucus gland, and cartilage, right, in the bronchiole and alveoli, right? And then, of course, don't forget the smooth muscle and elastic fiber. Smooth muscle and elastic fiber will be found in all the airways. Hang on. So this one will be found in all the airways, okay, until bronchiole. When it comes to alveolus, there is no more smooth muscle because you want the wall as thin as possible to shorten diffusion distance for gaseous exchange between the alveolus as well as the capillary. So that's why there's no smooth muscle. We only got elastic fiber in alveolus. Okay, so we continue to look at more structures now. So this is a micrograph that shows you a section of the trachea from a young man. So if it's from a young man, that means this young man already passed away. So that's why they can do the autopsy and then they can actually take some tissues out and make slides out of that. Okay, so what you can see here is that you can see that the ciliated epithelium layer, all right, it is actually pseudostratified and columnar. So this is a typical structure that you can find in trachea, right? So after that, the rest of them are connective tissues already, but the connective tissue has got different composition in different locations, okay? So basically, connective tissue will be made up of gel-like substance as well as fibers. So the fibers that you can find, it can be collagen, right? It can be elastic fiber. And then sometimes you also will find smooth muscle. Okay, all of them are found inside the connective tissue. Okay, so it will contain gel-like substance plus fibers. Okay, where's my collagen? Collagen fibers, elastic fibers, and then the smooth muscle. Okay, collagen fibers, elastic fiber, smooth muscle cells. Okay, so other than that, so I'm going to show you where you can find those things. So here, you can see that this area here looks like there are a lot of collagen kind of fibers. It could be smooth muscle also. I cannot really distinguish because it's low power. If I'm able to zoom in here at high power to check, then I can actually verify this wavy structure that I'm seeing here, which is pinkish color. Is this collagen or is this smooth muscle? I can't tell at the moment because I can't really see clearly, right? So I can say that it can be collagen, it can be smooth muscle. Unless I zoom in and check that inside they have got a flattened, elongated nucleus with spindle shape, but then I can verify it's smooth muscle. At the moment, I can't, okay? Now, so in this region here, can you see that this flowery structure, all this flowery structure are the mucus gland that you have learned, right? So mucus gland. Mucus gland. Take note of the spelling mucus gland. Huh? So it's flowery, okay? All of them are mucus gland. Now, I have not seen the duct. If I can see the duct, I will actually show you, right? So the duct, which conveys the mucus out of the gland, is actually round, okay, in a cross section. And then, of course, there must be a lumen, isn't it? So other than that, the rest of the cells will be either cuboidal, like in this case here, the cell is considered as cuboidal, right? I better draw, looks more like cuboidal. Okay, like this. Okay, so in a smaller duct, the cells lining the lumen. In this case here, it's called epithelial cell because this duct will go to the airways and connected to external environment. So these cells are epithelial cells. So they are cuboidal, okay, for smaller duct. Now from smaller duct, the duct can actually become bigger and bigger, right? So the bigger duct then, can you tell me what will be the shape of the epithelial cell in the bigger duct? Januli?
Teacher, can you repeat the question?
So I'm drawing a bigger duct now. Okay, this is a smaller duct, this is a bigger duct. So a duct is a tube that conveys the mucus out of mucus gland. Okay, so I show you that a smaller duct, the wall, which is made out of epithelial cells, the cells are cuboidal in shape. For a bigger duct, can you tell me, so what will be the shape of the cell?
Is it columnar?
Yes, correct, columnar, because if the duct is bigger, definitely the cell must be taller, isn't it? Right, the cell is taller. So it's no longer cuboidal, so it's taller cells. So those taller cells are called, the shape is called columnar. Okay, so they are columnar epithelial cell. Okay, all those cells here are columnar epithelial cells of the duct. So this is a smaller duct, right? This is a smaller duct and smaller. Okay, smaller duct. And this one at the bottom is actually a bigger duct. Okay, bigger duct. All right, so here I cannot see the duct, so I cannot tell you where are the ducts. Okay, oh, I think I can see the ducts. So the flowery structure here are the glands. The duct is this one here. I think I can see this one. Can you see this circle here? Can you see this circle here? Can you see this circle here? So this is mostly the duct, okay, because it's totally round. It's not like flowery kind of structure. And there is a lumen in the center, but it's very tiny, the lumen. Okay, and it's surrounded by one layer of cells. So this is the duct. Okay, so other than the duct, you can also see something that is spherical, round. So this one is the blood vessel because the lumen has got red blood cells. You can even see the individual red blood cells. Okay, when the lumen has got blood or red blood cells, and you check the cell lining the lumen, okay, lining the lumen, facing the lumen, the cell's nucleus will be flat because those cells are the squamous endothelial cells of the capillary. So all blood vessels, right, the cells lining the blood vessel lumen will all be squamous. So that's why the nucleus is flat. Can you see all the nucleus? This nucleus here is flat here. This nucleus is flat. This nucleus is flat and elongated with tapered end. So this is a blood vessel. This is another one. This is capillary. Okay. So this one also another one here. So it's a lot of blood vessels. A lot of blood vessels everywhere. This one also looks like blood vessel to me. This one. Okay, everywhere. Then the last part here, again, this is connective tissue, and they label it F because there are a lot of fibrous connective tissue. That means you've got collagen fiber, elastic fiber, and so on. So exactly what fiber we have to zoom in and see. Okay, so usually the pinkish color one with irregular shape are the collagen fibers. And then for elastic fiber, we need to stain it in order to see, or else to be totally transparent and colorless. Then if you want to see, you have to zoom into high power to check whether you can see something that's wavy with similar thickness throughout, but it's not stained and not pink color, then that is collagen. So unless you use a special stain to stain it black color, then you can see. In this case, we can't see because it's not stained. But it's not stained, the fibers, elastic fibers are not stained. Okay. So next one is here. So this looks like zoom in already. And there is a cartilage, which is very thick here. Okay, so let's look at this part here. So this part is, you can see the nucleus arranged in different layers. So it's pseudostratified, columnar ciliated epithelial cells. So this is connective tissue with a lot of, all these are the glands. Okay, all these are the glands because the lumen are empty. If the lumen got got blood like this one here. So this is a blood vessel, I think, but I have to go and zoom in and see one. This one could be a blood vessel, but I have to zoom in and verify, okay? So this one looks at the duct. Can you see not? Okay, these are the glands. Okay, and then this whole big piece will be cartilage. Whether it's trachea or bronchiole, I will not know unless I can see the entire tube to see whether it's C-shaped or fragmented. Okay, and then usually at both edges of the cartilage, you will find smooth muscle cells. Like here, the smooth muscle cells are quite clear, right? All cells are arranged parallel and got the nucleus is flattened, elongated with tapered ends. So this is a smooth muscle layer, very clear, but the smooth muscle layer here is not very clear. So I need to zoom in and magnify this region to double check whether there's smooth muscle. If there is, if I can see at low power, I must draw this layer as a separate layer and label it as smooth muscle. Because in plan drawing, you're trying to draw different layer of tissue. So every single layer of tissue, you need to be able to identify and to draw into your plan drawing. Okay. So here you can see all the cells inside the cartilage. So those cells, as I've said yesterday, they are located inside an empty cavity, or I call it the cave, the empty cavity, which is called the lacuna. One lacuna, many lacunae, and inside each lacuna, it can house usually about 2 to 3 cells, which we call chondrocytes. Okay, chondrocytes. About 2 to 3. Sometimes you can find 1. Okay, sometimes you can find 4 also. So it depends. You have to check the actual slide. Okay. So this is a higher power. So you're zooming in already. So the structure becomes bigger because it's higher power. So you can see this is also pseudostratified because you can see nucleus on top here, nucleus at the bottom. So pseudostratified, ciliated columnar epithelial cell. Okay. This part here looks like a different kind of tissue. Right, and it looks like there's a nucleus here as well, right? So this one could be smooth muscle cells, could be smooth muscle cell because the nucleus is flat and elongated, okay? Then this one is the cartilage, right? So again, so this is lacuna. The entire lacuna is filled with the chondrocyte. It's only one chondrocyte here. Nucleus, you can see nucleolus, and you can also see the heterochromatin at the side of the nucleus. So every detail you can see nucleus you have to draw, right? Okay. These are the rest of the cells in the connective tissues. Do I have smooth muscle? Can't see clearly the smooth muscle, this part here. Okay, can't really see that it's a smooth muscle unless I can find a nucleus that is flattened, elongated with tapered end. Then I can guarantee that, okay, that is a smooth muscle. Okay, so in that kind of case, what I will do is that I will be moving the slide around to check other regions when the structure is clearer to check whether it's smooth muscle or not. At this region, I cannot really see. I can't really tell. Okay. So this one is to show you that in the epidermis layer, we can also find where this is not goblet cell. Goblet cell. See this one with a lot of purple color dots inside it. So all these fat cells, very, very fat looking cell. It's not a fat cell. So these cells which look very big, with a lot of mucin droplets, mucin droplets. Okay, so they are the goblet cells. All right, so mucin droplet is found inside the goblet cells, right? Mucin droplets. So you can see that those goblet cells at the top here is actually no cilia one, right? So the rest of the cells got cilia, and not only that, can you see that at the base of the cilia, in this region here, it's got darker purple color. Now, this is actually the cytoplasm of the cell, but this region is darker purple compared to this is pale purple, right? So do you remember why this cytoplasm here, where cilia is attached to, is darker in color? Li Pei?
Teacher, I'm not too sure.
Okay, I open to the floor. It's to do with your chapter 1. You learn about structure of the cilia, right? What is the structure below the cilia called? Open to the floor. Hello? Okay, Ong Shu Min. Ong Shu Min? You all forgot already, huh?
Oh, not too sure.
Okay, this one chapter 1, huh? Okay, let me retrieve the PowerPoint so that you remember. Anyone remember or not? Okay, organelle structure and function. Okay, I open and show you, huh? Watch it. Hang on first. Hey, why it move like that one? Huh? Hang on. Okay, this cilia, right? This one. Okay, I'm not going to explain. You tell me why at the cytoplasm at the base of the cilia is darker in color. Based on this diagram you've learned in your chapter 1. What is the structure that causes the base to be darker in color? Type your answer. Anyone? Basal body. Lao Huyen says correct. It's basal body. Okay, Megan, correct, huh? So basal body, because whenever there is cilia, right, inside there must be microtubule, and the microtubule at the base is arranged like the centriole, which is 9 triplets, whereas inside the cilia itself, the microtubules are arranged in 9+2 arrangement. So that's why this one here, which is 9 triplet arrangement, causes the base of the cilia to look darker in color. Okay, so that's why the base is darker in color here. Can you see not all the base here? They're darker in color because of the basal body. Okay, so do you draw this as a separate layer? Yes, you draw them as a separate layer. As I've mentioned, the examiner already said, any part, any structure have got different color, you can draw a line to separate them, even though both are cytoplasm. That's what the examiner says. Okay, so since it's CIE, we follow CIE, right? Even though, never mind. When I learned in my uni, it's actually not like that. Okay, so whoever marking your paper, you will write in the way that they want you to write. Okay, now here can you see the basal body region is darker color. Then this is pseudostratified columnar ciliated epithelial cell, right? Okay, now I want to stress this again. When you call pseudostratified, that means it is a false multi-layer, right? Pseudo stands for false. False multi-layer. Okay, now I just want to say that when it's false multi-layer, it's because the cells are actually grounded right at the bottom, all of them, whether you see them short or you see them tall, right? They are all attached to the same level. But this cell here, I read the book yesterday, it says that this cell might not stretch all the way to the lumen. So it could be short, but because both of them are attached to the same base, right, same level of the base, so we are not considered this as multi-layered. That's the reason. Okay. So this is just a drawing to show you very clearly like where is a ciliated cell and where are the goblet cells. And goblet cells, you can see that usually this entire cytoplasm facing lumen will be filled with mucin droplets. So nucleus will usually be found below the mucin droplets. So this region here. And then the whole cell will look like Y-shape. Remember when I asked you to draw the cell is Y-shape because this region here is the cytoplasm, right? So this region here is cytoplasm, right? And then this region will be made up of mucin droplets already. So it's Y-shape with nucleus in the middle of the Y shape. Okay. Here you can see that in this drawing, very interestingly, the student actually draw a capillary. Can you see now? Another capillary here, another capillary here. The rest are the fibers and the cell that produce those fibers in the connective tissue. Okay. So this one is, okay, so this one is the ciliated epithelial cell that has been brushed out from the epithelium layer so that you can see that the nucleus is big. So when it's big, usually the examiner describes it as prominent. Prominent means it's big, so it's actually easy to see. Okay. And then the base of the cell looks like it's actually tapered. So it's got tapered shape. Taper means narrower, tapered ends. Okay, at the one end is tapered, means narrower. Narrowing is called tapered. Okay, so this diagram shows you the lumen and the two walls surrounding the lumen. And then you have got the pseudostratified again, columnar ciliated epithelial cells. And then we've got the goblet cells. Can you see goblet cells at the base? You've got the nucleus, right? These are the cytoplasm of the cell, mucin droplets. Okay, so this one is special because I think the mucin has been stained pink color. So that's why you can see the color here. And then the mucus, even the mucus on the surface of the lumen is also stained pink color. Okay, and the nucleus is stained purple color. Right, so the nucleus, this is a very clear goblet cell. Where you can see the cytoplasm, right, can be represented by two closely spaced lines all the way until the end. So this is the cytoplasm of the cell. So where do you stop your drawing? If you're asked to draw, the plan drawing is that you need to draw from here, right? Enclosing the purple color line, which is actually cytoplasm of the cell. So follow as closely as possible. Okay, I'll draw until here. Then this one, you have to guess where will the cell end because you cannot see, right? So you have to guess where the cells end. Okay, undo. So I cannot see, so I guess, okay, probably the cell will end here. So draw like this. Then another line is here, right? Okay, and then draw the nucleus, but following the shape of the nucleus. So this is called the detailed drawing that is accurate representation of actual micrograph. Okay, continue to draw the whole thing, right? For mucin droplets, you have to draw the mucin droplets if you can see individual droplet. Okay, you cannot use dot. You have to use any dot. You have to enclose the dot. Okay, and then you can draw them this way. Okay, so you can base on the shadow and you draw according to the shadow that you can see. Right? Sometimes clear, sometimes not clear. Okay, next, so this is scanning electron micrograph of the epithelium layer, which shows you the cilia as well as the mucin droplets, okay, from the goblet cell. So this one, again, repeat. So this section here is, you can see nucleus and different layers. So it's actually pseudostratified columnar ciliated epithelial cell. Okay. This one are the thick elastic fibers and it's stained. So you can see this is longitudinal section of a capillary. This is a transverse section of an arteriole. Why? Because outside this, there's another layer that form the wall. It's not only the endothelium layer, there's got another layer that form the wall. So this is, and it looks quite thick. So it's an arteriole. Okay. These are smooth muscles. Very obviously, this is smooth muscle. So we can know that this is smooth muscle. This one could be elastic fibers in between smooth muscle, right? And this elastic fiber is running. It's being cut transversely. So it's running this way. You can see, no? Okay, it's running this way. It's not running this way, right? So it's transverse section. Then you've got the cartilage here. Okay. So this one shows you there could be goblet cell here, but the nucleus is not visible, right? Or probably this is the nucleus of the goblet cell. It's got two cells here. Then this is the obviously ciliated epithelial cell. Okay, obviously ciliated epithelial cell. So you draw up all the way until wherever you can see. Then you stop there. Okay. This one is bronchiole. Bronchiole, it is said that approximately 1 mm, right? The thickness of the bronchiole is approximately 1 mm. So you can imagine how tiny is the bronchiole, right? And then it's folded so that when the air goes in, it can stretch. Okay, it can stretch. So the typical structure of bronchiole is very easy. You've got the ciliated epithelium. Usually is cuboidal, okay? And then you've got this smooth muscle ring around the wall. So the rest are connective tissues. That means collagen, elastic, fiber, elastic fiber, and the cells that produce those fibers, okay? So bronchiole, again, because there's no cartilage, okay? So obviously you can see the ciliated epithelium, okay? And then the smooth muscle and a lot of blood vessel, okay? A lot of blood vessels all over the place. Blood vessel, blood vessel, blood vessel, okay? Right. So this is the alveolus. So you end up in alveolus. So alveolar wall is very thin because it's supposed to allow a short diffusion distance between the gas in the alveolar space and the blood in the capillary. So the wall is very thin. So the wall is only containing whatever that's necessary for gaseous exchange. So you can be asked to draw detailed drawing of a wall of the alveolus. So you need to learn how to identify the different structures in the wall and draw them accurately. All right. So you can see that the alveolus on the left and right, and they got different color because they are prepared with different stain. So different stain will give different color for the same structure. So this is something that I want you to be aware of. Doesn't mean red blood cell will always look red color in the slide. It depends on the stain that's being used. Okay, doesn't mean that the nucleus will always look blue color in the slide. It depends on the stain that's being used. So on the left side, I want you to see that all the blue color are the nuclei, so they're all stained blue color, and red blood cells are pink in color, okay, or magenta. But on the right side, you see that all the red blood cells are dark blue or blue-black color. All the red blood cells are blue-black color, all this, okay, blue-black color. And the nucleus is purple color. Okay, the nucleus is lighter purple color. So let us look at the structure of the wall. So the wall must have capillary. So where is the capillary? All capillary got lumen. So same as the lumen of the alveoli. So you see this is white color, right, which is alveolus. You look inside the wall, if you've got white color similar to this, it must be a lumen, but cannot be of alveolus. So it is of the capillary. And inside the lumen, you expect to see red blood cells flowing in it. So this is the lumen, and you've got red blood cells here, maybe 1, 2, 3, 4 here. Okay, and this is the lumen here with the red blood cell 1, 2, 3, 4, 5, roughly about 5. So this is entire lumen. Then what about this light purple? So what is enclosing the capillary? Endothelial cell, right? So this light purple color must be cytoplasm of those cells that form the wall of either the capillary or the wall of the alveolus. So this is a cytoplasm of either the endothelial cell of capillary or epithelial cell of the alveolus. Okay, do I know which one is which one? I may not know. Okay, I may not know. Okay, why? Because the wall is very thin, but for here you might say that this belongs to the capillary, but for here this could belong to the epithelial cell of alveolar wall. Okay, sometimes we do not know one, but you must know, okay, this nucleus, this capillary, this red blood cell. Okay, so this is a nucleus here. You can see the nucleolus and the heterochromatin very clearly, and definitely the lighter purple must be cytoplasm of the cell. And beside here, you see this white color is a lumen of the capillary. This is red blood cell, lumen of capillary, red blood cell. This is a wall of capillary made out of cytoplasm of the endothelial cell. Okay, so that's how you identify the structure. So if you're asked to draw the wall of the alveolus to show the distribution of the structures in it. Okay, so these three are drawing from the student. Okay, drawing from student. And this will also be your homework. I do not wish to see any student just copy answer from another student. I wish you to practice your skill. If this is a micrograph given to you, or if this is a slide given to you and you're observing it under the microscope, do you know how to make an accurate biological drawing out of that? That is a skill that I want you to grow, not copying another person's answer. Please get it clear. Okay, Because if you copy answer, when you come to exam, you have not practiced the skill, how we're going to draw it nicely, and how do you know that you're confident you can draw and score full marks in exam? So do not ever copy any answer, because that's not the skill that I want you to practice. All of you know that. Okay, you pass up to me, very nice drawing, also no use, because you have not practiced it. So what's the point? Okay, I'm very angry because when I see students submitting answer, it's actually copying answer. Afterwards, when I discuss, you will know. Okay, I'm quite angry to see those kind of answers. Okay, because you're not practicing what you're supposed to be doing. So defeat the purpose of the homework anyway. Okay, so let's see. So among these three drawings, the best is actually this one at the bottom here. Why? Because it's very accurate. Accurate in terms of what? So when you're only asked to draw the wall here, right? So you have to draw this wall very big in the space that's given to you so that all the details can be drawn very clearly. So that's why your drawing must be big, occupying more than half, that's 3/4 of the space given. So you see the nucleus that's being drawn is so accurately drawn, which is the nucleus here. The nucleolus is shown, heterochromatin is also shown. And then beside the nucleus, there's this capillary here. Can you see this capillary? Look at the shape of the capillary being drawn. And the shape is the same. Can you see or not? And then this is the red blood cell and the red blood cell. Again, the shape, the size, the proportionality very accurately drawn. Then from here to here, you see this lumen got a line separating them. So the student actually draw this line separating these two lumen. Okay. And then this is actually a nucleus. This is a nucleus. So the student actually draw the nucleus here. Right at the bottom here, there's another capillary. See here? So another capillary, right? And then inside there, they've got cells. So the shape of the cell is like this. So the student draw like this, exactly what you see. And then this is another capillary here. Can you see or not? With the lumen. So the lumen is actually shown very clearly. So this is the lumen. And then this thin purple color line is also shown here because you can use two lines to represent the thickness of that wall. Okay. And then individual red blood cells drawn accurately according to the shape, the size, the location, everything. So that's why I said this drawing is the best. Accurate drawing. See here, even the red blood cell, you see the shape is exactly as what you see in the micrograph. And then the capillary, the lumen of the capillary is also shown. So this is the best drawing among all the drawings. Okay. So the rest of the drawing here also okay, acceptable, but not as accurate as this one here. As you can see, some things missing, or the shape of the structure does not really represent the actual shape. So not so accurate, but might be able to score full mark. It depends. Here is wrong already. How can the red blood cell be flowing inside the wall? It's supposed to be flowing inside the lumen of the capillary. So you need to use your theory to help you to make a correct drawing. Okay, so this will be your homework. Pass up next week Monday. Draw the wall of this alveolus. Okay, and also you need to label. You need to label. Okay, capillary, nucleus, red blood cell. Okay, so this drawing here, can you see this drawing is very different from previous one, right? A lot of details are not there and it's not accurate. A lot of things are missing. Right, compared to this one here. Can you see the big difference? Right, there are 1, 2, 3, 4 red blood cells, and here they've got zero red blood cell. So this is not an accurate representation. We'll lose mark in exam, definitely. Okay, so up to now, you should be able to describe the structure of the human gaseous exchange system, right? Including trachea, bronchus, bronchiole, and alveoli, and then the capillary. And then you need to include the microscopic structure of the walls of the trachea, bronchi, bronchioles, and alveoli. And describe the distribution of the following in the trachea, bronchi, and bronchiole. So where can you find cartilage, ciliated epithelium, goblet cells, smooth muscle cells, and capillaries? And of course, for alveoli, there will be no cilia. There is squamous epithelial cell, but no cilia. Okay. And also be able to state the functions of ciliated epithelial cells, goblet cells, mucous gland, cartilage, smooth muscle, elastic fiber, and squamous epithelium. So elastic fiber, you have learned the function in the blood vessel. So what is the elastic fiber function in the alveolus, for example? So let us look at this past year question. Describe the role of elastic fibers in the wall of alveolus, 2 marks. So you know that elastic fiber is a rubber band. So when the air flow into the alveolus, the wall of the alveolus will have to expand. So because of the elastic fiber, the wall can expand. So from this shape, from this size, when the gas go in, it may become bigger. So it will stretch. Okay, so when it stretch, what is so good about stretching is that stretching actually increases the surface area. You imagine this is the original surface area and now it's bigger, so it's got higher surface area. So higher surface area will actually increase, higher surface area will increase the diffusion rate, right? Rate of diffusion, okay, for gaseous exchange between the alveolar space and the capillary. Okay. So another advantage of this is, so more air can come in. So when the alveolus can stretch, higher volume of air can come into the alveolus. So better ventilation, right? And then after stretches, when you breathe out, you relax when you breathe out. So why? Because when you breathe out, all your intercostal muscle and diaphragm will be relaxed, and the elastic fiber is the one that will recoil to push the air out from the alveolus. So that's why it's relaxed. So the mark scheme answer is, you want the alveolus to be able to stretch on inspiration, which is inhalation, and recoil back on expiration, which is exhalation. Okay. So when the wall stretches, it will increase surface area of the wall, or it will increase the volume of air that can go in. For what purpose? To increase rate of diffusion or rate of gaseous exchange. Okay. So recoiling of elastic fiber will help to expel or force the air out during breathing out. And the stretching of the alveoli will prevent the alveoli from bursting when the gas go into the alveolus. Just like balloon, you don't allow the wall to stretch, right? It will break. Okay, if it cannot stretch, then it will break. So that's all for today for the gaseous exchange part. Okay.